Hello and welcome to class. So this is part 5 of the immune system video lecture series. So we're going to go through a scenario where a white blood cell like a macrophage could encounter an antigen. And this is a very nice flowchart actually that summarizes the process of what happens when our body encounters a pathogen with an antigen and what happens in terms of the white blood cells that are macrophages. So the macrophage would then be activated and the macrophage would digest the pathogen that had the antigen on its surface, a surface protein, and then that macrophage would display that antigen with its MHC marker for helper T cells to be activated then. Those helper T cells would switch into high gear and would divide into memory helper T cells that were specific for that particular antigen that the macrophage displayed. Remember, there are 3 antigen-presenting cells in our immune system response. And those types of cells that are antigen-presenting cells are the macrophages, the dendritic cells, and the B cells. So once that helper T cell is activated, and then it has produced your memory helper T cells for that specific antigen, that helper T cell would divide into an effector helper T cell. And that helper T cell would then stimulate and activate naive cytotoxic T cells to start dividing for that specific antigen. And we would have memory cytotoxic T cells produced for that specific antigen and effector cytotoxic T cells produced. That effector helper T cell would also secrete cytokines to activate naive B cells into active B cells. And those active B cells would because the helper T cell will display that antigen to them, they will then divide and form antibodies, specific proteins to bind with and fight that antigen in a large amount. The plasma cells can function for a few weeks producing 2,000 antibodies a second that are specific for that particular antigen. So that would help fight that infection. Those B cells that are now activated would also produce memory B cells that would be able to have that protein code, the antigen code, so they could produce antibodies with their membrane-bound antibodies that would be specific for that particular antigen to fight that antigen. So let's take a look at this one more time, this pattern of defense. So first we have a macrophage encountering a bacterium, a pathogen with an antigen on it. That macrophage is going to digest that bacterium and then take the antigen off of the bacterium and hook it to its MHC complex, that self marker, and produce MHC number 2 complexes, which display both the ID tag for our tissue cell to identify that macrophage as self, but also the antigen that is invading us. So then that macrophage would encounter a helper T cell, maybe in a lymph node or in the spleen, or even floating around in our bloodstream, and show that helper T cell the MHC and antigen complex, that MHC number 2 complex. At the same time, that macrophage would secrete cytokines, a type of chemical signaling protein that would activate that helper T cell and alert it to the threat. So the next stage would be alarm. Now that helper T cell is activated, and so the appropriate B cells and T cells to destroy that specific antigen are going to be activated. When activated, these cells divide to form clone cells that are designed to accurately and specifically fight that antigen. So this clonal selection is a very important part of our adaptive immune response. So the fifth step in our specific defenses is the B cells would be forming the plasma B cells or effector B cells that are going to be short-lived, but they're going to pump out that specific antibody at about 2,000 antibodies per cell per second into the bloodstream that would then be binding to the antigens. And then the T cells would also be activated to form cytotoxic T cells that can then attack specific body cells that have been infected with that particular antigen. So in part of the viral replication cycle, a virus binds to our cells and then invades that cell, gets into that cell, causes the viral DNA to be incorporated into the genome of that cell, and then causing new viruses to be produced with the viral proteins and the viral DNA or RNA, whatever the virus is using for their genetic molecule. So one thing that the cytotoxic T cells can do is act like specific assassins that can attack those infected body cells so that they can be disabled and killed before new viruses could be released. So in the sixth part of the immune response, now we have the antibody-mediated part of the immune response, where antibodies are used to fight and disable those pathogens. Antibodies have about 4 different ways that they can fight and disable a pathogen. They can do precipitation by forming these antigen-antibody binding complexes that can clump and are easier for the phagocytic cells like your macrophages and your neutrophils to engulf. They can also do lysis. So the antibodies can activate the complement proteins that can then form attack complexes so that the target cells will then burst open and die before reproducing that particular pathogen, like if it's a virus that is replicating within that cell. They can also attract phagocytes by marking them. So the antibodies can bind with the phagocytic cells. The antibodies can attach to the antigen on the bacteria and mark that bacteria cell for destruction by the phagocytic cells, by calling those phagocytic cells over. So basically like a tag, a little molecular tag. They can also do neutralization. So antibodies can bind to and surround even molecules like toxins in our body or viruses and cause them to not be able to interact with the proteins on our cell plasma membranes of our tissue cells, so that that virus would cause damage to the tissue cell. So here you can see a variety of antigens displayed, like on this bacteria cell, and then the antibody that would interact with that particular antigen. Here's an antigen displayed on a virus particle, and then an antibody that would be specifically interacting with that antigen on the virus particle. So here's our fourth step of alarm. So you can see now we have the helper T cell activating the B cell. The B cell that would then be able to identify that specific antigen, because B cells have membrane-bound antibodies. So now that B cell can produce those specific antibodies that would be able to fight that pathogen. And here you can see the memory B cells being produced that could fight that specific pathogen. So an antibody is a Y-shaped type of antigen receptor protein. It's made only by B cells, and it binds only to the antigen that prompted its making. Antibodies can activate complement. They can facilitate phagocytosis by tagging cells for or tagging the bacteria cells, for instance, for destruction, and they can neutralize the pathogens or toxins. So the structure of an antibody is a Y-shaped type of protein. And there are regions within that Y-shaped protein that can bind to the antigens. Part of the protein is constant, and then there are other regions that are able to interact specifically with the antigens. There are 5 classes of antibodies or immunoglobulins. So these are the 5 classes. And these are the different types of categories of antibodies here. So we're not going to go into a lot of detail about these 5 classes, but one good way to remember them is by thinking of the name MAGE, because that spells the classes, the M, A, D, G, and E. So different categories of antibodies are relevant for different types of immune responses. But these are major categories of antibodies. So the next part of the immune response is the cell-mediated immune response. In the cell-mediated immune response, we have effector cytotoxic T cells that are able to release perforins and proteases. And these are the enzymes that can then pop the membrane, the cell membrane of those cells that have become infected with a certain virus. So cytotoxic T cells are able to touch kill these cells that display the antigen MHC markers. And they can cause apoptosis, then programmed cell death, to deal with that pathogen. And here you can see a cytotoxic T cell killing a cancer cell. So that helper T cell would be able to activate the cytotoxic T cells and send them like little cellular assassins after the very specific cells that have been infected by that particular pathogen, like in the case when we have our body cells, our tissue cells, infected by a certain virus. Cytotoxic T cells can also identify cancer cells because they would be displaying different proteins on their cell membranes than a normal healthy cell would. And T cells, cytotoxic T cells, can also attack cells when we've had like an organ transplant or a tissue transplant, because those cells would not be displaying our normal proteins for our tissue cells. So we have continued surveillance because of the memory system of our memory B cells and our memory T cells and the adaptive immune response. So this is where we will start next time. Thank you.